DNA replication occurs inside the nucleus, specifically during S phase of interphase from mitosis or meiosis. The two original DNA strands are antiparallel. So this is 3' because it doesn't have the phosphate. This is 5', and then the opposite strand is antiparallel, so this would be 3'. It doesn't have the phosphate, and this is 5'. It does have the phosphate.
Helicase will unwind and break apart the hydrogen bonds between nucleotides. Primase will place an RNA nucleotide primer on the DNA.
DNA polymerase III, the primary enzyme in DNA replication, will start to put new DNA complementary nucleotides to build a new strand. However, it can only be built in the 5' to 3' direction. So since the new strand has to be antiparallel, and this is 5 to 3, the opposite would be 5' here to 3'. So this will be the leading strand, because it can go with the direction of the helicase and only require one primer. This side, since it's 5' to 3', the antiparallel would be 5' to 3' here, and it would have to, the DNA polymerase has to move in this direction, opposite of the helicase, creating a semi-continuous build, and it will create fragments of DNA known as okazaki fragments. So this is the DNA over here, putting in new nucleotides.
And then Primase comes and puts
Then on the lagging strand, it'll go in the opposite direction because it has to go 5 to 3', which is this way. And it'll also put in new DNA nucleotides from the primer.
So on the lagging strand, the first enzyme, because of the
DNA polymerase I.
Helicase will continue to break down the hydrogen bonds in the DNA. Primase will again put in a DNA primer, an RNA nucleotide up here because this is the lagging strand. However, on the leading strand, it can continuously build with DNA polymerase III new nucleotides.
We have to stop. I can't do this. I can't. I wish we can we like just do the like this is? I don't know. I can't do this. She's so I can't. I like actually can't do this. Can I get splice on my part? It like gets them all together. I'm gonna get splice. Oh my!
Now on the lagging strand, it can build this direction, the DNA polymerase III, going 5' to 3'.
DNA replication is the process. Now, DNA polymerase III two other ones that do not have the nucleus of all cells. As the corresponding
First enzyme in this process. The first enzyme in this process is helicase. It breaks apart the hydrogen bonds and then leaves the DNA.
The lagging strand has to go back in the nucleus and start
but it can't get
using RNA primers. So the DNA is antiparallel.
like that.
The leading strand can
Once the DNA polymerase III has reached the other RNA primer, it won't connect. This creates okazaki fragments, which are just fragments of DNA, because, well, it's a direct result of being the lagging strand.
DNA polymerase I will replace the RNA primer nucleotides to a DNA nucleotide.
So U will go to T over here. And ribose will change to deoxyribose. And then over here, it'll just change from U to T.
Finally, ligase will seal gaps between the primer and new strand growth, as well as the okazaki fragments.
So ligase will seal this between the two okazaki fragments and the primer, and also up here because this was a primer. And then only once in the leading strand, right here. And these are the two replicated complementary DNA fragments.